Spaniard. Amen, Donny and Sal. You guys are the best. Keep up the good work. Well, I appreciate that. I appreciate the kind words. So I got a few minutes here to address briefly some of these follow-up questions that have come in. First one, Geo plays piano. Do you have something to say about the chromosome 2 fusion? Haven't seen anyone on the YEC explain that. Yes. If you go to my website, standfortruthministries.com, I've written at least 2 articles on the chromosome 2 fusion. One article going over a general counter to the idea that there was a fusion. My second article is a response to Gutsick Gibbon, and I go over our findings. Chris Roop and I, we discovered in many other chromosomes, specifically the chromosome 9, we found similar signatures that you find in the chromosome 2 of these interstitial or internal telomeric-like repeats, TTA, GGG, CCC, TAA. The challenge has been to creationists, where do we find, other than the chromosome 2, which they assert is the result of a fusion event in the past in the human hominin line, they've asked, where do we find those telomeric repeats in other chromosomes in a head-to-head manner? And so we took up that challenge a year and a half, possibly 2 years ago. Time flies by when you're having fun refuting evolutionists. And we discovered, confirmed prediction. We assumed, we did expect to find other instances in the genome of these interstitial telomeric sequences that we do know have a functional role in the genome as they are binding to transcription factors and basically initiating a gene to be copied into RNA. So it works in the transcription process. And we found a fairly large signature in the chromosome 9, and it's even more pristine than what we find in the chromosome 2. And nobody would say that the chromosome 9 is a result of a fusion. And so the point is, we find similar signatures that we find in the chromosome 2 that evolutionists say, because of these signatures, this must have been a fusion, but we find similar signatures in other chromosomes. There's a long debate there. I've had formal debates on this issue. The biggest point is that the fusion argument, I just put out a video 2 days ago, so I recommend people check it out. The chromosome 2 fusion argument, it doesn't really matter because it's agnostic at the end of the day. Even if there was a fusion, creationists can explain a fusion in the human line, because even the evolutionary community admits that the fusion would have occurred after the hypothetical split between humans and chimpanzees. Appreciate that, Brother Doki. There's one of the articles. And so even they admit that this would have occurred after the split in the human line. There's evidence suggesting that if there was a fusion, it has nothing to do with human evolution, that humans and chimps are not related. There's a paper that talks about a specific type of heterochromatin, this densely packed kind of DNA that comprises a lot of repetitive sequences like satellite DNA. This is found lacking around chromosomes, and that includes the chromosome 2 of your chimpanzee and also gorillas. And so the lack of heterochromatin there suggests that if there was a fusion, at best, it would have been a fusion directly in the human line, but humans being unrelated to chimpanzees or any other form of life. There's a major problem for the evolutionary model, because if there was a fusion millions of years ago after the split, that would be a near extinction event. That person who experienced the fusion, they would basically be the sole ancestor of all people today, because today the 46 chromosome signature is fixed. Every single human being has 46 chromosomes. What about humans that once had apparently 48 chromosomes, or hominins? Okay, so that means the unilateral mistake occurs. This person has 46 chromosomes. What about every single other person on the planet at that time, or hominin? To the evolutionists, when and where did this fusion take place? Was it in an Australopithecine? Was it in Homo habilis? Was it in Australopithecus sediba? Was it in, we want some sophisticated answers. But regardless, you'd have other tribes, other people groups, other so-called hominins with 48 chromosomes. Okay, so for the 46 chromosome signature to become fixed, that means what? That means all people with 48 chromosomes would have to be wiped. All those lineages would have to be wiped out. They'd have to go extinct. They'd have to die in order for the 46 chromosome signature to proliferate. And so there's a problem in the evolutionary model accounting for the necessary fixation of the fusion. But in the creationist model, we have two bottlenecks. We have the flood bottleneck with 8 people, Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and their 3 wives. We also have the Tower of Babel event. Before people spread out to all parts of the globe, people at that time are confined to one geographical region for the most part. And so that could explain the proliferation or fixation of the 46 chromosomes, because either a subgroup within the pre-flood world got this fusion. Fusions are manifestation of genetic entropy or genetic degeneration. They're not moving things forward. And so a part of that group could have been Noah's family. So they go onto the ark with the 46 chromosomes, and then it spreads that way. Or sometime within the couple hundred years after the flood and before the Babel dispersal event, that fusion takes place. But because people don't spread out yet to all parts of the globe, it can become fixed before people spread out. And so point is, whether or not there is a fusion, it's agnostic to the overall debate. So quit using it, evolutionists. It doesn't help your position. It doesn't provide us with discriminatory evidence for evolution or for creation. But at best, if there was a fusion, the creationist has the better explanation. Creationists can account for it. Evolutionists like to say, well, we predicted it. We expected it. Yeah, because there was an inconsistency there, because humans have 46 chromosomes, great apes have 48. So there must have been a fusion of some kind. And we discovered a possible fusion. Again, there is some evidence suggesting that there may not have been a fusion. But again, it doesn't matter, because if there was a fusion, it's agnostic to the overall debate. And so it's a good question. And that is an answer in a nutshell. But I have done several debates. I've also given several talks and lectures on the topic. And I recommend people to check that out. Geo Plays Piano says, genes can be repurposed. Well, that's your assertion. That's your evolutionary philosophy. Okay. In relation to ERVs, show me a non-functional endogenous retrovirus. Show me, as a matter of fact, an exogenous harmful retrovirus like HIV. That's an exogenous retrovirus. Show me that infecting somebody. Okay. And then infecting in the right way, where it's passed on vertically. Vertical transmission to become an endogenous retrovirus. Now it's occurring from within. Okay. Now that person has the endogenous, the offspring has the endogenous retrovirus DNA. Show me how mutations now wipe away the harmful effects of it, the harmful attributes of it. And then show me how that non-functional ERV goes from non-functional to something as incredibly functional as embryological development, where without that ERV, we couldn't exist. That's not even my words. That's from the conventional literature, that apparently without ERVs, mammals and humans wouldn't exist. And so why do we share ERVs with other forms of life? Well, why do all modes of transportation have engines in common, or batteries in common, or tires in common? This is good design. And when evolutionists say that we have 200,000, that's actually a misconception, because we've only got a few full ERVs, as in you've got your LTRs on either side. Your full ERV does share sequence similarity to a retrovirus, an exogenous retrovirus, where you've got the LTRs on either side. That's long terminal repeat. Then we got GAG, POL, and ENV. That's your envelope protein. We've got those genes. That's an ERV. The majority of so-called ERVs that we find shared between humans and chimps, they're solo LTRs, where you don't have the LTR, the GAG, the POL, the ENV, the LTR. No, it's just solo LTRs. That's it. And so it is assumed, it is asserted that these solo LTRs are degenerated ERVs, where they were once part of the full ERV sequence. The problem for the evolutionists, though, is the fact that these solo LTRs are actually serving as binding sites for initiating or enhancing gene expression. And so the better interpretation is that these solo LTRs are not degenerated ERVs. They simply represent functional stretches of DNA. So sure, we can share thousands of solo LTRs with chimpanzees, but because of their functional roles, this is just good design. The better answer is common design. The evolutionist has to show we don't want just evolutionary philosophy and imagination and story time. We want to know. We want to see empirically how these so-called ancient leftover remnants of past exogenous retroviral infection can become functional in determining cell types, in acting in embryological development. We know they function in the immune system. We know they function in tumor suppression. Notice this paper. Switching sides, how endogenous retroviruses protect us from viral infections. Long disregarded as junk DNA or genomic dark matter, endogenous retroviruses have turned out to what? Represent important components of the antiviral immune response. That means these ERV units are like antiviral programs in a computer. You need them. This is good design. And so all of the biological world, let's take primates, for example, or chimpanzees, they also require functional DNA units that fight off bad viruses, bad viral infections, invaders, okay? And so the fact that we share these with the chimpanzee is no surprise at all. Notice this. They not only regulate cell immune activation, but may even directly target invading viral pathogens. Here's an interesting point. In order for ERV and ERV-like sequences to carry out their operational roles, they have to look like the retrovirus in order to fight it off. And so, of course, they're going to share sequence similarity with the retrovirus, because to carry out those roles, they have to look like the retrovirus. They perform viral mimicry, for example. There's papers that go into the current research, cancer research, where transcribed ERVs, GAG, POL, ENV, LTRs on either side, the full ERV, they transcribed ERVs in a cell. They give that cell the appearance of being invaded by a harmful retrovirus, a harmful virus. And so the immune system, working with what's called the P53 protein, this incredibly designed, highly sophisticated protein called the superhero or guardian of the genome. This protein works with the immune system to target that cell more destruction, because it looks like it's being invaded by a retrovirus, but this is a functional designed ERV that's performing viral mimicry to get the immune system to destroy that cell, that tumor cell. This is incredible. But guess what? When the evolutionist says, well, why do ERVs look like retroviruses? That must mean they were once retroviruses. These must be the ancient remnants of past viral infections. No, they require the sequence similarity to retroviruses to carry out their functional roles. If they didn't look like the retroviruses, they couldn't perform functions like viral mimicry. It would be like a police officer or FBI agent infiltrating the mob, but showing up undercover with their FBI name tag or their uniform. Okay? They're not going to do their job very well. Okay? They're going to be caught. They have to look the part. So they would have to come in disguise. They have to look the part. And every single property or component of the ERV is necessary to carry out these roles. For example, the ENVs, the envelope proteins, they function in blocking harmful viruses from entering the cell. And they do this by binding to very specific receptors that infectious bad viruses might use to enter the cells of hosts. This is amazing design. Okay. And the same thing applies to the other aspects of the ERV. Again, the LTRs, the GAG, the POL, the ENV, these proteins that are heightening or amplifying immune responses through the binding of these receptors. And so how could these functions come about in the evolutionary paradigm? Randomly inserting viral DNA into the genome of a biological organism will more likely disrupt genetic functionality rather than improve upon it. If I were painting a picture and I were to put a blindfold on and start randomly applying brush strokes, am I going to make that painting any better? No. I'm going to more likely make it worse. Okay, so randomly inserting viral material into the genome of an organism is not going to one day work towards gaining a function in embryological development or determining cell types. Geo says, okay, but aren't ERVs from infections? Well, the interpretation of ERVs being from infections is because they share sequence similarity to exogenous retroviruses. But the conventional literature is showing that the many functional roles associated with ERVs are dependent upon that sequence similarity to ERVs, to retroviruses. And so that overturns the major reason why evolutionists believe that ERVs are from retroviral infection in the past, vestiges of a retroviral infection. Now, here's the thing. The majority of ERV and ERV-like sequences are fixed between humans and chimps. Humans do have some unfixed, where only a subpopulation have them. Well, here's another interesting function. We found this in a paper studying koala bears and their ERVs. Okay. Those pre-existing designed ERVs, they actually function because right now koala populations, they're being bombarded with exogenous retroviral infections, and the pre-existing ERVs, they're functioning to disrupt the endogenization process of those exogenous retroviruses. That's incredible. So they're fighting off these retroviruses, not only from invading, but if they do invade in the right way, they'd have to infect a germ cell line and be passed on. These pre-existing designed ERVs can actually disrupt the endogenization process. Okay. But just like how we have these incredibly designed DNA repair enzymes that fix DNA breaks, it is said that in a single day, in a single cell, and we have 65 trillion cells, that you can experience up to 1 million DNA breaks. 1 million DNA breaks. But thank God we have these incredibly designed DNA repair enzymes that fix most of these breaks. Now, unfortunately, they don't work perfectly. And so not every single DNA break is fixed. That means many are passed on, right? The mutation rate in autosomal nuclear DNA is roughly 100 to 200 per person per generation. But those DNA repair enzymes, they're designed, they're functional. Okay. They're very important to the health of biological organisms. They do their job great, but they don't do it perfectly. And so, yes, we do have some unfixed ERVs that are most likely due to true exogenous retroviral infection. Okay. But these are unfixed. We'd predict that they're largely non-functional, at least in the sense that they're necessary to sustain life, like embryological development. Okay. And that is because the designed units of ERVs that fight off exogenous retroviral infection, they work good, but that doesn't mean they work perfectly. And so it's perfectly reasonable that occasionally, the system being flawed and not perfect, an exogenous retrovirus could infect in a germ cell line and be passed on to the next generation. But again, we could make a prediction as a creationist that the majority of those would not be fixed, and they wouldn't have the kind of functions that we see in your fixed ERVs, where they're necessary to sustain healthy life processes. But the question for the evolutionist, Sydney Raptor, let's ask you, Mr. Evolutionist. Okay. What is the origin of retroviruses? Retroviruses require a host to replicate. Okay. So what came first, the host or the retrovirus? If the retrovirus came first, where's the host? Where's the cell for it to replicate? That's why even in the conventional literature, you have what's called the escapee hypothesis, where retroviruses result or originate from within the cells of genomes, within the hosts. Okay. Errors in the copying process or recombination or so on and so forth can result in a retrovirus. And then from there, that retrovirus, which contains those components that are necessary for things like retrotransposition and infection, moving from one host to another. Okay, they have those properties now. So if they were to jump to another host, the possibility is there that since they have that pre-existing ability to do things like fight off other viruses or act in the immune system or act in cell stress responses, there's a possibility that they could take that pre-existing ability from one host that they originated from, probably from a pre-existing functional retrotransposon. They could take that pre-existing ability into a foreign host. Because viral DNA is selfish, they might want to protect that host from other viruses coming in and disrupting the function or health of that host. And so that possibility is there. But when it comes to a function like embryological development, no. I challenge the evolutionists. I challenge Sydney right now. Show me naturalistically, evolutionarily, what's the origin of retroviruses, and show me a non-functional endogenous retrovirus gaining a function in embryological development, where without that ERV, we wouldn't exist. Okay, so Sydney Raptor, let's see what his answer is. Unlike many, I don't, okay, so that's a non-answer. I don't claim to be proficient in biology and genetics. Yeah, well, here's the thing. That's a real shame, because it's in genetics where we answer the question of ancestry. It's genes, traits, and genetics that are inherited sperm and egg. Not a fossil, not geography, not geology. And so the best way to answer this question of ancestry is through our genetics, and you lack answers to important foundational questions that work towards answering this greater question of ancestry, where the evidence is pointing to what? Creation. It's you guys that have put forth these arguments from junk DNA, pseudogenes, and ERVs, your so-called best evidence for evolution. But what we're finding is that these so-called evolutionary leftovers or viral DNA, these genetic mistakes, they have incredible functional roles that you evolutionists can't account for. You also have this paradox or this chicken and egg problem for the origin of retroviruses. But we have, as creationists, and this is consistent with what we find in the conventional literature, that retroviruses have originated from host genomes. And that makes sense because retroviruses require a host to replicate in the first place. And so Sydney Raptor, you lack an answer. So any evolutionists in the chat, show me how these functions could have originated naturalistically through selection, through mutations, through recombination, whatever you want to appeal to.